Assalamu alaikum wa rahmatullahi wa barakatuh. Welcome to a new video. Today's lesson is about one type of specific immune response, which is cellular immune response. If we want to summarize the defense lines against invasion by the pathogen, or we want to understand them more, we can say that they are like walls. Okay, we have the first line of defense against pathogens is the natural barriers, which we talked about earlier in another video, and then we have the second line, which is the inflammatory response or the non-specific immune response. So the pathogen have to cross these walls, the pathogen has to cross these walls every time it wants to enter the body. Now, what if the pathogen succeed to cross the second line of defense and the non-specific immune response fail? There will be a third line of defense. But what is this third line of defense? It is the specific immune response. How is this specific immune response activated? First of all, we talked about the inflammation or the non-specific immune response, and we took an example of if we injured ourselves, our skin was cut, and a pathogen enters the skin, a reaction is called inflammation reaction takes place, which is a non-specific immune response, and a process called phagocytosis takes place, where phagocytes like monocytes or granulocytes try to engulf the pathogen through several steps and then eliminate it and try to engulf other pathogens also. Now, at the end of this process, phagocytosis, the macrophage, the monocyte or the granulocyte starts a process of activation of specific immune response where several steps happen. First of all, this phagocyte, okay, presents the antigen of the pathogen on its surface, okay, the antigen fragments, as if it is calling for the immune system and giving the information of the antigen that it just swallowed and digested. So the macrophage, okay, after ending phagocytosis, it presents the antigens of the pathogen on its surface, and now it is called an antigen presenting cell. So this macrophage now is called an antigen presenting cell. Now, for whom does this antigen presenting cell present its antigen? There comes the T helper lymphocyte, if you remember, a lymphocyte called T helper or T4, which has certain receptors on its surface called antigen receptors. These receptors bind to the antigen present on the surface of the antigen presenting cell. Okay, so the T helper takes this antigen on its receptor, and now the T helper is activated. It's turned on. It was as if it was asleep, and now it wakes up and starts its action. What does this T helper lymphocyte do? As we talked earlier, the T helper lymphocyte is important for the control of all the immune response. It is responsible for activation of other T lymphocyte, the other T lymphocyte, which is the T killer lymphocyte and the B lymphocyte. But how does the T helper decide which lymphocyte will it activate, the B lymphocyte or the T killer lymphocyte? According to the type of the pathogen. So the T helper, after it receives the antigen from the antigen presenting cell, it has to study this antigen, know its type, and activate either the B lymphocyte or the T killer lymphocyte. What is the criterion? First, the T helper lymphocyte, if the antigen is a circulating antigen or a molecular antigen, it will activate the B lymphocyte. What do we mean by circulating antigen or molecules? For example, some types of bacteria, toxins, etc. And in this case, when the T helper lymphocyte activates the B lymphocyte, a humoral immune response takes place, which is one type of specific immune response. But if the antigen is what we call a cellular antigen, okay, the T helper lymphocyte will activate the T killer lymphocyte, or another name for it is the T cytotoxic lymphocyte. Cytotoxic means it kills cells. What is example about cellular antigen? Like what? For example, cells infected by viruses or bacteria, So the cell, when it's infected by a virus or a bacteria, it becomes an antigen, it becomes a non-self. It will carry the antigen of the virus or the bacteria. It will no longer carry the MHC of the body. So it will be considered as an enemy. It has to be killed. Same as for the cancer cells or the tumors, which also, due to the mutation of the DNA inside them, they become non-self. They will carry a new surface antigen, where they will become non-self, and they have to be fought and killed. And finally, grafted cells. Grafted cells, transplants, for example, if we take a skin from another donor, if we take a kidney, a liver, any other grafted tissue or cell, it might be considered a non-self body, and it has to be fought. So these are examples about cellular antigens. The specific cell to kill or fight cellular antigens is the T killer lymphocyte. Okay, now if this antigen or this T killer lymphocyte is activated, the response is now called cellular or cell-mediated immune response. So due to the type of antigen, cellular antigen, the response is called cellular immune response or cell-mediated immune response. Now, what is the mechanism of cellular immune response, or how does it take place? What happens during a cellular immune response? First of all, when the T helper lymphocyte is activated, it has not only choose the T killer lymphocyte or the B lymphocyte, it has also to choose the specific T killer lymphocyte. We have in our body hundreds of T killer lymphocytes. Each T killer lymphocyte is specific for a specific or a certain antigen, a certain cellular antigen. So a T killer lymphocyte, for example, is specific for a certain virus. Another T killer is specific for a type of bacteria that enters the cell or infects a cell, or for a certain cancer, etc. No, so there are many types of T killer lymphocyte, many specific T killer lymphocyte. So here, for example, this is an example. We have two killer lymphocytes, 1 and 2. The difference between them is their receptor. Notice the shape of the receptor on their surface. The T helper lymphocyte has to decide which T killer lymphocyte will it activate, and when it finds the T killer lymphocyte specific for the antigen, it activates it using interleukin. It secretes certain types of chemicals called interleukins and activates the specific T killer lymphocyte. So it will not activate T killer lymphocyte 1, it will activate T killer lymphocyte 2 due to the shape of antigen. Second step, when the specific T killer lymphocyte is activated, it goes to the infected cell. This is the infected cell that displays non-self antigen. This antigen is non-self, okay, as if it is calling for, it's saying that I'm infected, I'm non-self, come and kill me. I have a different surface marker than my neighboring cells. So the T killer now binds to the non-self antigen on the surface of the infected cell, and now the T killer lymphocyte is activated. It's turned on. Okay, it will start its action. After that, after the T killer is activated, it starts to secrete certain chemicals that are found inside sacs. There are sacs inside the T killer lymphocyte that carry certain substances, for example, perforin. Okay, the T killers start to secrete this perforin, okay, and this perforin start to eat the membrane of the infected cell and create pores inside it. So now the infected cell membrane is perforated, it has openings in it. Okay, so again the T killer lymphocyte secretes perforin, one type of chemical, okay, that makes pores in the membrane of the infected cell. Now, after this, the fluid surrounding the infected cell will enter quickly to the cell through these pores, and this will cause the cell to explode, to lyse. Okay, so the fluids that are surrounding all these cells, our cells are all surrounded by fluids and ions, they will enter quickly due to the difference in pressure. The pressure outside the cell is very stronger than inside of it. They will enter quickly, and the infected cell will explode just like a balloon. Okay, it will be lysed and it will be dead, of course. Finally, like any other dead thing in our body, we have to clean it up. The macrophage comes again. Now it has another role to clean up the site. It has to eat and phagocyte all the dead or lysed cell to prevent infection and inflammation, to stop inflammation and to prevent formation of pus, etc. Okay, so here we notice that the phagocytes or the phagocytosis is a process present in several places, not only during non-specific immune response. I hope you understood the lesson today. Thanks for watching. Make sure to come back for new videos on immune system. Goodbye.